Hello, welcome back to Educator.com. Today we're going to discuss the present day problems or challenges in the world of microbiology. And these aren't the only ones by any means. There are just two very important ones. And it isn't that we're not going to discuss these concepts all the way through the course, because we will. But these are very focused discussions to really drive home the point that the field of microbiology is exploding as far as expanding the frontiers of biological science. So the first concept that we really want to discuss is the gut microbiome, and so much work is going on in this area. It's just phenomenal, and it's really been, I would say, the last 5 years that there has been more and more recognition about the importance of the bacteria in the human intestinal tract. And then the other concept we're going to discuss is antibiotic resistance, which is becoming a real challenge certainly in the healthcare environment, but also just in human health in general. So to start us off on the gut microbiome, this is really, even considered to be an organ in and of itself. And, there is so many ways that these gut bacteria interact to either promote and enhance health, or certainly to promote the onset of disease, and sometimes even to make recovery of disease even more challenging. And we're continually finding new things out almost on a daily basis. So if you can imagine our digestive tract consisting of all these different populations of bacteria, and these bacteria really vary in relationship to where we live, in relationship to the types of food that we eat. Do we eat meat? Are we vegetarian? What sorts of general patterns of food do we eat on a daily basis? All of these things contribute to the different types of bacteria that live in our digestive tract. So just to be to discuss this in a more detailed way, you can see by this figure here that, in a healthy gut environment, we have a population of different species of bacteria that live in a commensal way. And what that means is that basically it's a friendly relationship where the human host is basically allowing these organisms to live in the digestive tract, and these are not human organisms. These are foreign cells, right? They're not human cells, and yet the human immune system is tolerant to these organisms. It doesn't eradicate them, for instance. And so they live in this commensal way with the immune system. And, if there is an infection by an intestinal bacteria, for instance, in some cases, in a healthy situation, we can have a, increase in immune reaction. The pathogen causes, for instance, diarrhea or some illness symptoms for a day or two, and then it goes away, and then the digestive tract resumes its commensal state and everything is fine. But there are other factors that can really serve to alter the balance of this relationship. Things like antibiotics, of course, we learned in the and we will certainly discuss in the future the role that antibiotic resistance plays, and that's the second topic of today's lecture, for instance. So antibiotics certainly can alter the composition of the gut environment. Different foods change the composition of the bacterial environment. Hygiene. Do we live in a sanitary environment? If you go to developing countries, the sanitation in some cases is challenging, and that the folks who live in these environments have learned over time that their bacterial population in their guts, for instance, have learned to tolerate an environment that folks raised in a very hygiene type environment, for instance, can't survive in for very long. If you go to a country where you're not used to the bacterial populations, you, for instance, need to boil the water, you can't eat some of the food, and if you do, one can get very ill from that. So this onslaught of these various environmental factors, different pollutants, viruses, for instance, can create a condition called dyssymbiosis, where there is a decrease in the so-called peacekeeping bacteria and an increase in these pathologic bacteria. And when that happens, you can actually lose the integrity of these intestinal epithelial cells, which, in the healthy state are very tightly connected to one another. But in this pathologic situation, you can actually develop gaps in the intestinal epithelium, and then you can have leakage of fluids, nutrients. So, for instance, if you have these bacteria living in the gut environment and you have a leakage, then perhaps, you're providing even more nutrients to these pathologic bacteria. But also these bacteria can actually cross the epithelial barrier and enter the bloodstream, for instance. So this damaged epithelial barrier is significant. It can increase these pathologic bacteria in relationship to their adherence and penetration. And then you can get a pathologic inflamed condition. So some autoimmune diseases like Crohn's disease, for instance, is a disease where, for some unknown reason, the commensal bacteria are recognized as being foreign, and then the immune system will launch an attack as though these bacteria were pathologic, and then you get this autoimmune situation with Crohn's that can be very serious. So this altered state of immunity isn't just restricted to the gut environment. It can also affect your susceptibility to other diseases. It can affect whether or not, for instance, you're obese. So one of the contributors that's thought to promote weight gain and obesity now is the composition of the gut environment in relationship to these bacteria. So we see that genetics certainly plays a role in effective, having a strong immune system and a strong immune reaction. The environment certainly influences how suppressed or how vigilant the immune system is, for instance. So things like stress and diet and infection, vaccination, all of these things influence not just our capacity to protect the human host from enteric pathogens, from intestinal diseases, but also respiratory diseases, skin infections, bloodborne pathogens, all of these things. And we know, for instance, if we have a healthy, strong immune system, we can withstand quite a lot of pathogenic insult and really not show any symptoms at all. So really, the gut, the bacteria that live in the gut are turning out to be a huge new frontier of research that many investigators are beginning to evaluate and pay much more attention to. And so throughout the course, we're going to really discuss ways in which we can potentially promote the health of the gut microbiome in relationship to protection from some of these other diseases. And certainly it's becoming increasingly important, especially in the face of this antibiotic overuse and development of antibiotic resistance. So just to review, that the gut has thousands of different species, a mixture of these commensal beneficial species of bacteria that if you have an unnatural shift in this balance, you can actually cause microbes that are normally benign to become pathogenic. You can also increase one's susceptibility for development of other diseases. You can cause chronic inflammation, and chronic inflammation has its own set of autoimmune processes that happen as a result. So these pathogenic microbes that are pathogenic under certain conditions are also known as these pathobionts, if you can imagine. So they've sequenced the gut microbiome in relationship to these other biomes, these other microbiomes, and they're not really that different. But the interesting thing is that they've looked at them in relationship to these metabolic pathways that groups of these genes are involved in. And you can see that there is some variation between individuals. So, for instance, based on where you live on Earth, you're going to have different populations of bacteria inhabiting your intestinal tract, just simply because you're eating different kinds of foods, for instance. So there's a number of different factors that can certainly influence what bacterial population you happen to have living in your gut. And these bacteria have their own genomes, right? Their own genes, and potentially these genes can synthesize various constituents where there can be an interaction between the human host and the gut microbiome in relationship to things like a good example is vitamin K. That vitamin K is produced by the bacteria that live in a very specific place in the colon. And if one, for instance, is undergoing an extreme type of antibiotic treatment, you can actually eradicate these bacteria that synthesize vitamin K. So, for instance, individuals sometimes when they have these IV antibiotics also need to go on vitamin K supplementation. So just by the fact that one eradicates the bacterial population in the gut, you can completely even enhance, for instance, your adverse reaction to a disease. So it's really important to manage, right, and promote the healthy commensal survival of these gut bacteria. So an example also is E. coli. Now, E. coli is normally a benign bacteria that lives in everyone's gut. It lives on our skin. It's everywhere. But occasionally these E. coli can become pathogenic. They can even produce toxins. The Shiga toxin producing E. coli, for instance, is a foodborne E. coli, pathogenic E. coli that can cause extreme forms of disease and even death. There was an outbreak in Germany in 2011 that was quite extreme. These pathogenic organisms were carried in meat. And so anyway, you can certainly have benign bacteria become pathogenic based on certain environmental changes and potential changes in the gut as well. So these genomes of bacteria are thought to encode 3.3 million genes, which could even supplant or outnumber, so to speak, the human microbiome or the human genome in relationship to determining how the body functions. Now, the other thing, though, that you must recognize that in science, when scientists get excited about something, it's almost like this, everybody jumps on the bandwagon for a while, and then, all diseases are related to the microbiome, and then there's usually a kind of resettling of the truth in the matter. And so we'll probably find out that the microbiome doesn't cause everything, but certainly it's related to quite a few health conditions and diseases. There's no doubt about that. So there's a great, if you're interested in exploring more about the microbiome and health, this link is terrific. It's an NIH website that provides a good introduction to some of the issues that investigators are evaluating in relationship to the microbiome. And there's there's a huge body of literature that's developing in this area. So this link will certainly lead you to some very good sources if you want to learn more about it. And certainly in this lecture series, we're going to discuss this concept quite a bit, especially in the bacteria lectures. Now, the second topic of present-day importance in microbiology is antibiotic resistance. And really, the concept of antibiotic resistance is based on the theory of evolution. So, yeah, sure, probably many of you have had the theory of evolution in your biology courses, and hopefully you haven't forgotten all of it, because it isn't just a theory that Charles Darwin came up with in the 1800s. It's actually a very important theory, set of laws that there are two areas in medicine right now where this concept, the theory of evolution, is directly applicable. One is antibiotic resistance. The other one is chemotherapy resistance. And in both cases, this natural selection, survival of the fittest concept, adaptation is really an essential principle to why we are seeing such a rise in some of these bacteria that in the past were not really much of a problem at all. So again, we will discuss many of those situations in the bacteria lectures that you'll find in the rest of the course. So just to recall the theory of evolution is based on the idea that populations evolve or change over time because those members of the population that are able to survive whatever adverse conditions in the environment, those individuals who survive will pass on their genes to their descendants, and that those traits that enabled them to survive will get carried on in the future generations. And Charles Darwin came up with this theory after many years of observation. And these theories are certainly proving to be true in relationship to antibiotic resistance. So if you want to know more about evolution, this terrific website, evolution.berkeley.edu, is everything you've ever wanted to know about evolution. It's an open source website. You can download anything you like. It's a very clear depiction of the role of evolution. And in fact, a number of the reference sources that I use or that are used in this course come from this evolution.berkeley.edu, the CDC, the NIH, Nature, the Nature publication series, New England Journal of Medicine. I really, various medical database sources. We've really tried to bring the top quality information into this course. So it's important for you to know that. And I think it'll come through as we go along in the various lectures of the class. So just to review what natural selection really is all about, it's based on the concept of differential survival and reproduction among individuals that vary in these shared and inherited traits, and that any trait that enhances an individual's capacity to survive are going to get passed on and through reproduction in a particular environment. So But when we discuss the application to bacteria, you're going to see that the bacteria have taken the natural selection concept to a new level, and we'll see how that works. So these antibiotic resistant bacteria are so let's back up a little bit, calm down, and discuss how this how do these antibiotic resistant bacteria come about. So we can take the example of a typical 2-week course of antibiotics that are basically designed to eradicate the particular bacteria that caused whatever symptoms of illness the person happened to present with. But what ends up happening is, because that agent is a little bit more generalized than that particular bacteria. So for instance, some of these antibiotics are targeted to gram-positive cell wall. So not only is the gram-positive bacteria in question eradicated, but other gram-positive bacteria that may potentially be beneficial or at least commensal in the digestive tract, those bacteria are eradicated as well. And these antibiotic agents can actually create this very extreme selection pressure that is equivalent to thousands of generations of environmental pressure that these organisms are used to. So because of this extreme pressure, we get the evolution of these antibiotic strains that occur quite quickly. They can occur within individuals. They can spread from individual to individual. They tend to be localized in hospital environments where a lot of individuals are either suffering from the same illness or are susceptible because of having other kinds of illnesses. So we're going to see again through the bacterial lectures exactly how and why this can become such a huge problem. So this is just a graphic. Actually, it comes from evolution.berkeley.edu, and you can see a population of bacteria, that some of which include resistant varieties, and then we administer these antibiotic agents. And one of the reasons why, for instance, as a patient, you're instructed to take the full course of antibiotics is so that you can make sure that you eradicate all the bacteria. Isn't that a great idea? But the idea also, or the thought is if you take the full 2 weeks, you'll eradicate every bacteria, that no resistant bacteria will be left behind. But what's happening is that is not occurring. So anyway, so we're left with this antibiotic insult. We have one individual there, and remember, some of these bacteria can self-multiply, but they can actually engage in another process that we're going to discuss in a minute. So anyway, so you get a increase in the population of resistant strains, and then by the end of the course of the 2-week regimen, you have a new population of all resistant bacteria. So the thought is, if you don't take the full regimen of antibiotic treatment, that there'll be some bacteria left behind, but there may be some bacteria left behind anyway because they were already resistant to the antibiotic agent. And with this evolution over time, it's even a higher likelihood that it's even more possible to have these antibiotic resistant bacteria, or at least be infected by them. So this natural selection process is enabling these populations, plural, of antibiotic resistant bacteria to multiply and replicate. But the other thing that happens with that bacteria, it isn't just that the non-resistant bacteria die and the resistant one live. Sometimes you can have bacteria in real time transfer for instance, their resistant genes to other bacteria who don't have those genes, and even before they all die out. So you get the resistant gene passed over to this bacteria that didn't have the gene before, and then pretty soon now you have more bacteria with this this antibiotic resistant. And so not only is it the generation survives, but you can actually have this genetic transfer that is occurring in real time, where it's almost like this resistant gene can spread to other bacteria, and that's another mechanism by which you can increase the numbers of resistant bacteria. So these acquired resistance is a major reason why these antibacterial agents are not effective. So we have the example of C. difficile, which is a gram-positive bacteria. And the nature of the bacteria, this bacteria has spores. So it isn't even that, you know, you pass like bodily fluids between patients. Because this particular bacteria has spores, what ends up passing from patient to patient are the spores. And these spores can live on surfaces, they can live on hands, they can live on just different equipment, they can even live on, you know, some of the tools used in patient treatment. They can live on surgical equipment. Now, of course, these are all sterilized, but things that aren't, that's why, you know, for a hospital that has a C. difficile infection, boy, that hospital really needs to be sterilized. So anyway, the C. difficile is becoming a huge problem in these hospital environments. So the resistance, it isn't just inheritance through descent. You can also pass these resistant genes to other non-resistant bacteria. So these highly resistant bacterial populations can live and flourish, in fact, in areas of high antimicrobial use. So this is a graphic, so to speak, put out by the CDC that provides some examples of how these antibiotic resistant bacteria spread. So one, of course, is by individual to individual, usually through these healthcare environments. So in other words, if you're a patient hospitalized, even for a non-infectious reason, certainly you can develop a bacterial infection while you're in the hospital. If you get, for instance, go in for surgery and then you develop an infection from that, and then through antibiotic agents that you're given, you can develop your own population of resistant bacteria that can spread within your family, within, you know, you go home after being discharged, you know, folks that you're close to. But also there is use of antibiotics in our food supply, especially in meat, that animals get antibiotics. They also develop resistance just like humans do. And you can potentially get antibiotics. You can actually have antibiotics in the meat itself, or the meat can serve as a vehicle for transmission of these antibiotic resistant bacteria. So it's a double way in which you can actually spread and cause transmission of these antibiotic resistant bacteria. So we're going to discuss several examples of ways in which, some very specific examples about ways in which we as a population, just worldwide, but also in the Western countries as well, where we're misusing antibiotics and we're actually creating the problem. So many hospital-acquired infections are caused by multiple resistant organisms, and there are 4 ways in which we are, as a society, misusing these antibiotic agents, and we're promoting this selection of these very resistant bacteria. So one way, physicians some physicians, not all, use multiple antibiotics. So, for instance, you come in with a serious respiratory infection, and you're given one antibiotic, it doesn't work, so you're given another antibiotic, it doesn't work, and so forth. And sometimes you're given 2 antibiotics, and you get a long course of therapy. And so what ends up happening is not only do you eradicate the pathogen in question, but you also eradicate the commensal bacteria. And it's an overuse situation, and sometimes it's not necessary. Now, you also are sometimes prescribed antibiotics, for instance, for a respiratory infection when the actual agent, the causative agent, is not known. So you come in with a fever, symptoms of a respiratory infection of some kind. Now, we will learn in this course or in this series of lecture that respiratory infection can be either bacterial or viral. So pneumonia, for instance, there are some pneumonias that are bacterial, and there are some pneumonias that are viral. And antibiotic agents do not work against viral infections. They only work with bacterial infections. And there's even a bacterial Mycoplasma pneumoniae that's a pneumonia caused by a bacteria that is not treatable with antibiotics, or at least antibiotics targeted to the cell wall, because they don't have cell walls. So anyway, what this is doing is this indiscriminate use of antibiotics. So for instance, if you're given an antibiotic and you have a viral infection, it's not going to do anything. It's not going to cause you to recover any quicker. It'll take away the bacteria, maybe if you have a joint viral and bacterial infection. But if you have a viral infection, if you have influenza, for instance, and you're given an antibiotic, it's not going to do any good. So this unnecessary use of antibiotics is promoting resistant bacteria, and these future antibiotic agents are not going to be effective for fighting that true bacterial infection. So, for instance, if you indiscriminately take antibiotics for every respiratory infection you might have, when the real infection comes along, the really serious bacterial infection, you may not be treatable, for instance. So this is really something that I think is very important to think about as if you're a patient or a health provider, for instance. The next reason is the ease in which we can obtain antibiotics. So in many countries, antibiotics are sold over the counter. You do not need a prescription. And this, again, encourages the inappropriate and indiscriminate use of these antibiotics. So if you're out living in a country where you can get antibiotics over the counter, you might want to try and evaluate whether or not indeed you do have a bacterial infection, and is it serious enough to warrant use of an antibiotic agent. Because if, for instance, you can get over it without using an antibiotic, it's much better actually to do that if you can, because not only are you saving the antibiotics for the true time that you need them, but also your immune system will certainly learn about this particular bacterial infection, and then you'll have naturally conferred immunity, which is, in my opinion, the best kind of immunity to have. Use of antibiotics in animal feed to prevent infections and promote growth. So at least in Western countries in the United States, for instance, we have these factory farms, chickens and eggs and dairy and cattle. pigs, and they can live in very close quarters. And one of the ways that the farmers prevent infection among these animals is to put antibiotics in the feed. And so these animals, like was shown in that figure, these animals can develop resistance to the agents just like humans. And those resistant bacteria can actually contaminate the milk. You know, we had that salmonella in the eggs in the United States. It was very challenging to identify where they came from. We can have a lot of different long-term effects from this practice. Now, some farms in the United States, at least, are moving towards this organic, non-antibiotic, non-hormone added type of farming, and unfortunately the cost of meat from those situations is much higher. But hopefully if more farmers change to this particular way of raising their animals, we may be able to take care of this problem to some degree. So there's a great tutorial on antibiotic resistance. It's kind of like a little time clock situation where you can watch how quickly these bacterial species can evolve. It's pretty dramatic, and it's not that different from how things are in the real world. So it's quite interesting if you have the time to look into this. So our first question, our first example for the day is, what do you think happens to intestinal bacteria when antibiotics are taken? Well, you have to think about which antibiotic agent is it. So we'll learn throughout these lectures that certain antibiotics target certain attributes of bacteria. So one well-known target, of course, is the cell wall. So these gram-positive bacteria, for instance, are given a particular regimen of antibiotics that target properties in the cell wall, and the cell wall of these bacteria are located on the outside of the cell, so they're an easy target. But all the other gram-positive bacteria that live in the gut, for instance, are also eradicated. So, I mean, it's a general practice in medicine now that when you're put on a regimen of antibiotics, it's often recommended that after that course of antibiotics, you take yogurt or kefir, which is a liquid form of yogurt, and many cultures in the world have different curd, or in the Middle East they have these different sorts of yogurt type spreads, and all over the world there are different types of these probiotic, which are actually bacteria that are living in yogurts and various dairy products and so forth. Sauerkraut apparently has these commensal bacteria, and now these you can buy pretty high grade these probiotic bacteria that are very helpful for the digestive tract. You can actually buy high-grade bacteria that come in these capsules. They're very expensive, but they're refrigerated. And if you see in your health food store, for instance, look for these refrigerated live bacteria in capsules. Those are much better to take. So in any event, it's recommended that after your course of antibiotics, you try to restore the commensal bacterial population, and you can do that through probiotics. But in general, to answer that question, what happens to these intestinal bacteria is that they get eradicated essentially along with the pathogenic bacteria. And the pathogenic bacteria could be even respiratory, for instance, but you're going to get your bacteria gut eradicated as well. So the second and last example for this lecture is, what are some of the things that we can do to reduce antibiotic resistance? Well, we saw those 4 examples where we are right now as a society, or as many societies on earth, we are promoting antibacterial resistance. So what we need to almost do is potentially reverse some of the things that we're engaged in right now. So, but again, these kind of things take years to implement. They take an entire change in society. We're so rooted in, for instance, factory farming. So we need to, lower the reliance on antibiotic feed, right? In the feed, right, of our livestock. Okay, so we need to decrease reliance. We need to decrease the overprescription. We need to potentially implement other creative forms of therapies. Now, we're going to talk about that in some of our bacterial lectures, is we can actually use probiotic treatment to potentially, if it's an enteric bacteria, that's a pathogen, for instance, we're going to talk about in the lecture in gram-negative bacteria, gram-positive bacteria, we're going to talk about Vibrio cholera, and we're going to see an experimental treatment where we actually introduce these commensal bacteria, these engineered commensal bacteria into the gut to basically crowd out the Vibrio. So use probiotic therapies to crowd out these other pathogenic bacteria. The third potential way is to teach the immune system to do a better job of eradicating and identifying, or first of all, identifying and eradicating the bacteria. So if our immune systems were doing a great job, we wouldn't need antibiotics. Okay, so teach the immune system to do a better job of identifying and eradication. So identifying and eradicating the bacteria. And finally, just keep pushing forth our frontier of developing new agents, right? New agents. I'm not even going to call them antibiotics. Maybe there's some new term for some new bioengineered method for eradicating bacteria, but finally, to increase our reliance on vaccination, right? So if we can lower the tendency for these bacteria to develop new strains, why are they developing new strains? They're developing new strains because of the selective pressure imposed by overuse of antibiotics. So if we can develop more effective vaccines, for instance, we also don't need to rely on antibiotics. If we can prevent the disease from even occurring, then we don't need to worry about use of antibiotics. So that concludes our third lecture on the modern day uses of microbiology. Thank you so much for visiting Educator.com.